shifting gears quite a bit. I'm going to talk about sensory abilities and the importance of understanding the sensory world of organisms for the next 3 lectures, I think. That's how long this is going to go. Probably today we'll mostly talk about vision. A lot of that's just because vision is so important to us, and so naturally we think of it first. So I want to tell you about these folks. This is Jakob von Uexküll, who introduced a concept which I think is incredibly important, and this is a tick. And these types of ticks. Back, I don't know, 18 something or other. What do we think about ticks? I mean, our perspective on ticks is that, females need a blood meal in order to mature eggs. And so what they do is they go out and they seek a mammal, okay, to feed on, and they get, suck blood, eat blood, drink blood, and get engorged with this blood. They drop off, mature those eggs, and basically lay them in a group, and then die, basically. Okay. Now, what's happening here in this situation is that ticks actually pay attention to 3 things, and only 3 things. Light, which is the first thing that they pay attention to. They use light cues to go up things, like up a grass stem or up a tree branch, okay, until they get to a place where they've reached the top of it. Okay. And at that point, they stop being sensitive to light. They don't pay any attention anymore to light. And instead, after the point where they get to the tops of a grass stem or a tree branch, they stop paying attention to light. And now they become sensitive to, and only sensitive to, butyric acid. The smell of butyric acid. Butyric acid is the thing that makes rancid butter smell rancid. Okay, that's the smell of butyric acid. This is a common component of the body odor of many mammals. Not really concentrated, but it is something which is detectable in sort of human or deer or whatever body odor. When they smell butyric acid, when they detect butyric acid, they drop off the branch. Okay, and now they're not sensitive to butyric acid at all. And they continue to be, have no sensitivity to light. Now they're going after a warm spot. Okay, they're sensitive to temperature and maybe a little bit hairlessness. So they seek out a warm, relatively hairless spot. Okay, and they burrow their head into that. Okay, and then if it's a deer or if it's us, there's blood that they can drink, and they drink blood, but they're not tasting blood. Okay, they don't have taste receptors at all. Any sort of warm fluid that you give them at this point is what they will drink until they're swollen up and full as a tick, and they drop it. And that's the world, the entire sensory world of a tick. Okay. They pay attention to 3 cues: light, butyric acid, temperature, and in that order. And once they get one cue fulfilled, they stop being sensitive to that and go on to the next cue. Okay. Everything around them in the world that they might be, have a pot that provide, the cues, all of the myriad cues of the external world, all the visual cues, they're not sensitive to any of those things. Any color signals that they get, they're not sensitive to any of that, the colors of things. They're not sensitive to the taste of anything. There are lots and lots of olfactory cues, chemical cues, that they might be sensitive to, but they're not sensitive to any of them except butyric acid. Okay. No sounds, they don't pay any attention to sound. The sensory world of the tick is very different from the kind of sensory world that we live in. I mean, they actually, it's hard for us to maybe extrapolate the idea of what the sensory environment of a tick is. What's cool? Let's see. Applied a particular term, which is really useful. Umwelt. It's one of these great German terms. And it is used to refer to the sensory elements of the environment that are relevant to the organism that you're studying. So you speak of the Umwelt of the tick, the world that it lives in, particularly the sensory world, the sensory environment that an animal lives in. I mean, this kind of gets a little bit back to my notion that we should be animal-centric, okay? I mean, we pay attention, what we're really interested in doing here when we're thinking about sensory abilities of organisms is trying to establish, what is the Umwelt of the organisms you're studying? What kind of sensory information do they have access to? How do they use it? What sorts of, and part of that is then what sort of things don't they have any access to or they don't pay any attention to? The Umwelt of an organism. Okay? And the way you think about what a tick is doing changes dramatically when you are informed by the sensory capacities of this organism, because the tick seeking out a mammal so that it can feed on blood is not exactly what is happening in this organism. It's engaging a particular set of sensory cues and doing certain things that really don't have to do with blood exactly, or mammals precisely, but they let the tick behave in a way that's kind of important for that organism. Okay, this Umwelt is the sort of overriding important thing for them. We're going to talk a lot about sensory systems, vision, hearing, electrical senses, olfaction, and so on. And a lot of the cases, the sensitivity of organisms, in particular sensory systems, is pushed to almost the limit that you can imagine. So, for example, in vision, the action of one photon can cause conformational changes in a sensory chemical. Okay, so you can't get less than a photon, in terms of sort of visual signals, visual information. In hearing, say in humans, because of the sort of massively parallel way that we have our hearing set up, we can detect vibrations in our inner ear that are far, far less in magnitude than the diameter of a hydrogen molecule. So really, really, really sensitive to very low sort of sound vibrations. Electrical senses, organisms can actually be sensitive to the movement of just a few ions in sort of electrical fields. Or in olfaction, organisms can be some organisms are able to detect just a few molecules and cause behavioral changes just by detection of just a handful, a countable number of molecules by their olfactory system. So one of the things that we'll see a lot are systems where, the sensory systems do a lot of averaging, or summing up of information in order to achieve some of these great levels of sensitivity. So you pay attention to this sort of thing here. So we're going to talk about vision first. And I'm going to actually kind of go to the board a little bit. Let's see. Let me go over here. Kind of build this up in sort of like, maybe not the most, I don't know, it's an easy to follow way, I think. So the first thing we think of in terms of visual capacities is the ability to see color, okay? And the extent to which we are able to detect color. And we'll also, a bit later, we'll spend some time talking about visual acuity, how organisms differ in the extent to which they are able to pick out details. And I'm not sure how far we'll get. We might get into polarized light detection, but we may not. Okay. So if we're starting off with vision, and starting off with color vision, I mean, the first thing we might think of is organisms that don't have color vision. They only have black and white vision. Achromatic vision, monochromatic, maybe is a better way of putting it, see black and white. Monochromatic, they only see, shades of gray, basically. This is broad vision only. This is not incredibly widespread. I guess it is some level of variety. I mean, we see organisms, things that we're familiar with in terms of the just black and white vision are like rats and mice are monochromatic, have monochromatic vision. Just take the levels of light, levels of hamsters. Raccoons and possums, sort of common around here. These are all nocturnal animals. Out abroad at night. Deep sea fish. Maybe that's not a surprise because there's not a whole lot of color down there. Some cetaceans, many cetaceans, have just monochromatic vision. Okay, and they, it's just the sort of activation of the rhopsin, that leads to their ability, or is associated with their ability to see. Okay, not so interesting. Now we have dichromatic vision. And here we're now into color vision. Okay, these are two different pigments. There are two of them. They're opsins, and they have different sensitivities to different wavelengths of light. So if we just kind of graph something like goes dichromatic vision, you have one pigment that's sensitive to, so these are of light. Wave length. G in there somewhere. Okay. In many cases, there's a lot of things that we're familiar with that have dichromatic vision. Most mammals have dichromatic vision. Dogs and cats. Ungulates have dichromatic vision. Squirrels, New World monkeys. There's some marine fish that do. I mean, what we're talking about here are two pigments that are, let's see, it's about bluish and then sort of greenish, maybe yellow, greenish yellow, something like that they're sensitive to. Okay, and the perception of color, there's two kinds of cone cells. There are those that have blue and those that have greenish pigments, and they respond or they don't. So it's digitized in that way. But the perception of color is a graded set of things. Okay, and perception of color has to do with the whether or not when they're exposed to a particular kind of light, it excites the blue cell and the yellowish cell at a particular frequency and rate. And so color is basically some sort of measure of the ratio of the responses of these two kinds of cells. Outside of these ranges, dark red, they may not be able to see that as a color because they're not sensitive to the pigments are not sensitive to that frequency of light. So dogs are not colorful. I mean, they're not without color vision, which is sometimes what you hear. Dogs or cats. They just have dichromatic color. They have two pigments. Okay. Now, I have to erase female because we're going to go on to trichromatic. Trichromatic vision. What? It's just instead of two pigments, it's one pigment or two pigments, there's three pigments. Okay, and it's not quite this, but as you get to some shorter wavelengths down here, to longer wavelengths, it's approximately blue, green, red. It's not quite RGB, but it's close to it, RGB. Okay, think of it. And again, the perception of color is not, there's only three kinds of cells. Those that have the blue or the green or the red pigment. The perception of color is something that happens in your brain where, some combination of green and red makes, yellow or orange, depending on how far you're shifted towards the red side of things. Here we have some familiar organisms because we're trichromates. Okay. Among the mammals, it's only the Old World monkey lineage, basically, that is trichromatic. Okay. And since we're, the apes come out of Old World monkeys, we inherited their ability to see, have trichromatic vision. Make sense? Now, here you think of, the wavelength of light going from blue to lower wavelength, a longer wavelength, sort of in a straight line. And similarly here, blue to red. But we often see the color portrayed in this sort of triangle, where would we red, green, and blue. So we can see combinations of colors, the things that we perceive as colors, within this triangle of sensitivities. Okay. You know, spectral colors, the colors that physicists see, or that, you know, that stars have, or something like that, go this way. They go red to green, green to blue. These are the spectral colors. These are the things that sort of exist. These are the wavelengths of light changing from longer to medium to shorter wavelengths, from lower to higher frequencies. Okay. If you break up, you know, white light with a prism, you end up with colors along this sequence. Okay. If you have, for example, though, some sort of pigment in nature which reflects light equally at blue and red, you know, ends, okay, that actually produces a color that we perceive. Okay? You mix together red and blue, and what do you get? Purple. Purple. But purple doesn't exist, you know, outside of our brains. I mean, our brains construct the color purple. It's not, it's a perceptual color. We can recognize it. We can be trained to distinguish purple from blue or purple from red, but it's not something that there, you know, you can find a wavelength of light that is constitutes purple. It just doesn't exist. Okay. If you combine light at all frequencies equally, what do we call that? Light. Light. And where's light? It's here in the middle. You know, equal combination of the response of our three sorts of cone cells to red, green, or blue. So that's kind of cool when you think about it that way. Now, who's trichromatic? In this way, old world monkeys, there's some freshwater fish that are like that, a few reptiles and amphibians that are trichromatic, but there's a whole group of other trichromatic organisms that are different from exactly our version of trichromacy. Again, three cells, receptor cells, that have particular, you know, sensitivities to wavelengths of light, but instead of starting with blue, we're going to start with UV, blue, and then a sort of a greenish yellow. Okay. Many insects, many spiders, many crustaceans are trichromatic in terms of their vision, but it's built upon the three, a different set of three pigments, a UV, the blue, and a sort of greenish yellow pigment. Okay. They also have a triangle of perception that goes from greenish yellow to blue to UV, and it's kind of, you know, not huge a distance into the UV. Okay. Again, the spectral colors are going from, you know, greenish yellow to blue to UV. Those are wavelengths of light that would cause this perception of color in, you know, a honeybee. honeybees can see UV. One thing that's kind of nice or interesting or whatever is that they don't have a red pigment. Okay, so they can't see red. This makes it actually, this is useful for us sometimes as scientists, because if we want to study the behavior of honeybees in the dark, because a lot of times they're doing things in the dark, we can just put them under red light, and they can't see. Okay, so they, because they can't perceive red, they treat it as the dark. Okay, so it's great. So, but there are possible situations where a honeybee sees a surface that is equally reflected in the UV, the shortest wavelength that it can see, and in the sort of greenish yellow. Okay? Over here. What is this? Well, it's something that's called bee purple. Okay? Because the idea is that purple is a combination of the longest and shortest wavelengths that you can see. Okay? And this is what bees see. So this is bee purple. What does it look like to us? Come on.
Regular purple?
Hmm?
Is it like regular purple to us?
No, because regular purple to us is a combination of red and blue, and they can't see red.
Is it green yellow?
Green yellow, because we can see that, but we can't see the UV, so we just see the green yellow part. But they see it as a different color. They see it as purple. And how do you know that? You could train bees to go to, feeding stations of a particular color. Okay, and originally, when they discovered that bees could see UV, is that they were giving bees two different cards, and they were constantly picking one of them as opposed to the other. And it turned out that the one that they were picking actually had UV reflection in it. And the investigators couldn't see this, but they figured out that bees could. Okay. So you can train a bee to come to a bee purple card and not to a green yellow card. Even to us, although to us, they look exactly the same. Does that make sense?
I was just curious, where would, on the trichromatic, where would black be, or nowhere?
Because there's no reflective light.
Yeah.
Yeah. What if you combine equally light as UV, blue, and green yellow?
White?
And let's call it bee white, because that's what it is. In fact, so what does bee white look like to us? I don't know, what's a combination of blue and green yellow, sort of blue-green, I guess. Because we can't see the UV component at all, it just looks kind of blue-green to us. But that blue-green color is different than if you additionally add UV to it, which for, we don't know what honeybees think when they see this, but they're getting equal contributions of all of the pigments that they can, colors that they can sense. So it's kind of white in terms of our definition of white. Okay. So I've done enough damage with my pictures. This is what the old world monkey human trichromatic vision sensitivities look like. It's not nicely evenly spaced. It's much more bunched up. There's a blue pigment and a green pigment and a yellowish red pigment or some orangish pigment. That's what it looks really big mammals. Okay? And our capacity to see red is when it's mostly responding to the color that we call red is when mostly this pigment that is responding to the longer wavelengths of light is providing most of the information. Okay. We're not done when you get to trichromats. Oh, here's what the real sort of color triangle looks like as you go from shortest to medium to longest wavelengths. You know, the longest wavelength is red, and the shortest is blue over here, indigo maybe. But you know, the combination of red and blue is purple. There are now also tetrachromats. Tetrachromats. Oh, I could draw this, but I don't want to do that. I want to subject anybody to that. Tetrachromats. There are 4 pigments. This is something that birds have. Turtles. Some butterflies. Freshwater fish. Tetrachromacy seems to have been the ancestral ability in terrestrial vertebrates. So we walked around on land as tetrachromats. Okay, the mammal lineage lost 2 of these pigments. Lost the UV and it lost the red pigment because most of them are dichromatic. And then in the old world monkey lineage, this gene that produces this particular opsin duplicated and moved a little bit towards the red. And that's why you end up with that thing. Okay. But this is a tetra. This is what the range of spectral sensitivity that tetrachromatic sensitive individual species with tetrachromatic vision see. Birds see in the UV. Okay. You know, not deeply into the UV, but a ways. You know, pay attention to 370 nanometers here. We'll kind of come up with that again in just a bit here. That means that bird vision doesn't exist in a triangle. It exists in a tetrahedron. Okay? As you go from the shortest to the medium to the long wavelength of light, it's kind of equivalent to what we can see. Then they add them in UV. So the entire range of spectrally real wavelengths go here from UV to blue to green to red. But they can see anything that's a combination of all of those responses by any of those pigments. So anything within that tetrahedron. And what that means, of course, is that, you know, birds have a much different and in many ways much more discerning capacity for color discrimination. You know, there might be certain colors that we see as exactly the same, but if you just add more and more UV into that, birds are able to distinguish that, you know, readily as different colors. Though to us, because we don't see that UV component, it's just down here in, I don't know, yellowish. Okay. Does that make some sense? So that's something, you know, and vision is going to be something that's terribly important when mammals or old world monkeys or birds, going from di to tri to tetrachromatic, are doing things like going through the environment, or trying to find food, or trying to hide, or trying to attract mates. So that's what the pigments look like in terms of their sensitivity. But in bird retinas, they also have these little oil droplets, which you can just barely see. They're circled in a couple of places here. And those oil droplets absorb light of particular wavelengths. Okay, so for example, like this is, the sensitivity of that particular cell to frequencies of light, but those oil droplets have absorbed a lot of that bluish light. Okay, and that sharpens the sensitivity range of the colors that birds can see, and it gives them a much greater sort of ability to, make judgments about color, discern different colors. Okay. Well, who cares that birds can see this? And then I guess the first answer is birds care. Some birds are sexually dimorphic. And in the case of birds, it's usually the case that if there's a colorful sex, it's males, and the less colorful sex are females. Okay, but not everybody's sexually dimorphic in terms of color patterns. There's a lot of birds that are regarded as sexually monomorphic. There's a couple of them. Some cases, the birds are pretty colorful, but they look the same. This is a blue tit. We'll see that in an upcoming video. This, I believe, is a male. This is a female. But they look pretty much alike. Here's a drab species. These are reed warblers of some species. And I can't tell which one's male, which one's female. They look exactly the same to me. But it turns out that relatively recently, somebody got the bright idea to actually take a look at the plumage colors of these sexually monomorphic birds under UV. And it turns out that about 90% of the bird species that we recognized as sexually monomorphic, because we couldn't see the difference, actually have different color pattern, or their male-female color pattern differences in the UV. So, for example, this Acrocephalus warbler, one of them, whatever the male is, has a bright, this eyebrow stripe is actually shines quite a lot, brightly in UV in males. And because we don't see it, we regard them as sexually monomorphic, but of course the birds are not regarded as sexually monomorphic. And this is, how much more important could that be to understand that there's differences between males and females. This is the myna, which is, in this case, it's being sort of, it has an iridescence to it. And starlings have kind of iridescent plumage to them. A lot of times when you have iridescence, that is a signal that there's probably some UV components to it as well. And this is kind of what happens when you look at it in UV. We'll talk about this in a second. Let's take a look at this video.
If we look closely at starlings, for example, we can see a sheen to them.
Whoops.
If we look closely at starlings, for example, we can see a sheen to their plumage, but that has an ultraviolet component that makes them appear much more vivid to one another. Blue tits, in our eyes, are one of the more colourful of our garden birds. But in ultraviolet, they're much brighter still. Their crests are particularly vivid and much brighter in males than females. So to them, these sexes look very different, whereas to us, blue tits all look the same. We would all, I imagine, think that budgerigars are unusually colourful birds, But ultraviolet radically changes the character of their costume. Their feet glow. And the spots on their cheeks, which are not particularly prominent to our eyes, positively blaze. Indeed, a Budgerigars full dress uniform is dramatic in the extreme.
There's a little bit of sleight of hand in this, but I'll just say that in case of blue tits, the females do actually seem to pay attention to the brightness of the forehead patch in UV, and they choose males depending upon which one has the brighter UV patch. It's sort of like business with the budgies and how they're glowing. We're seeing visual. We're seeing the color in the visible light. Okay, it's not like we're seeing UV light in this sort of like video clip. But what happens with parrots, and this is actually sort of a thing that they're doing, is that their feathers fluoresce in UV. So you shine UV on it, and their feathers kind of emit a visible light. It's that brighter kind of like pink, blue, white color to it. And that enhances the color in visible light. And that's what we're seeing. So we're not really seeing the UV portion of this. We're seeing what the camera is recording, which is visible light. Okay. So it's a little bit of a bait and switch, a little bit of a mistaken thing here. But cool anyway. Okay, so birds see a whole lot more colors than we do. Okay. Where's the buttons? Gotta love capitalism, because what people have actually tried to do is take advantage of the fact that birds are known to see UV, and you can buy paint if you're a duck hunter, so that you can paint your duck decoys so that they are actually a little bit more visible in UV light. So yeah, good way to make money off of bird vision. If we're going to be a little, I don't know, sensitive about our lack of visual abilities, when we know that birds can, or tetrachromats, let's take a look at stomatopods here. These are mantis shrimp. They don't have just four. This one, I think, they're measuring the spectral sensitivity of 11, by my count, different visual pigments in the eyes of mantis shrimp, which is interesting, number one. And one of the things is, I don't know, we said 370 was kind of the little peak for birds, so they're actually further into the UV here in terms of what mantis shrimp can see. Okay. But their eyes are actually built differently, and their brains are processing. It's not so much that the eyes are built different. The brain is processing this information differently in mantis shrimp than in, say, humans with trichromatic vision. So, for example, you can determine what sort of spectral sensitivity the mantis shrimp have, and it turns out to be about 20 nanometers. So, from 500 to 520, if they're different by more than 20 nanometers, they can tell the difference in colors. Okay? And the way you do that is, you train them to go for a food reward if it is seeing a 500 nanometer signal, and then you test it with 600. Oh yeah, I can just make those distinctions and pick the 500. 550 does fine. Get to 510, and you can't tell the difference between them. Okay. So it has about a 20 nanometer limit in terms of whether it can distinguish colors. Okay, which is you know, fine. Humans can do 2 nanometers. Okay, so we could do, you know, 502 in comparison to 500. So we have the way that we're treating that information is we're sort of like cognitively, you know, assembling the difference between the color patterns so that we can actually distinguish many more colors than Mandelshrim, who are not necessarily using whatever 11 chromatic vision or something like that. Or, the deca chromatic. Yeah.
What about like the opposite side of the spectrum, like infrared light?
I'm sorry?
What about the opposite side of the spectrum, like infrared light?
You know, so there are some things that see longer wavelengths. C is a kind of put quotes around that. For example, rattlesnakes and a few other snakes have capacity to detect heat, which is infrared. Okay, and they actually, boy, see if I can draw a rattlesnake again. Here's a snake head. They have a pit organ, or pit vipers, little thing. And when it's pointing forward, it is able to detect infrared from a potential kangaroo rat or something like that is out in front of it. Okay. They also have a little thing here, little thing that sticks up. It casts a shadow, so they can tell, you know, when the mouse is in the shadow of its pit, viper pit. Okay, so it is able to detect in total darkness the presence of, you know, a hot mammal out in front of it. Now it's not the eyes doing this, but the information that comes from the eyes goes to a place in the brain, and the stuff from the pit also goes to that same spot. So it's mapped onto its visual field. So it's kind of, so there's a limited ability to see infrared. Yeah.
Dr. Koh, could you please explain the part about the Mandelshrim again? If they have 11 humans, does that mean that they can see so many more colors than humans, or is it much closer?
It's actually because of how they do it. They're just looking at, you know, those cells that are responding to, I don't know, lost this, I don't know, yellow cell here that responds to yellowness. Okay, and they're not comparing necessarily the ratio of the response of yellow to the neighboring orange and the neighboring green. It's just whether or not it's yellow. So that doesn't give it a whole lot of ability to distinguish between colors. Okay, so they're actually not cognitively assembling the information about what a color is in their brain in the same way that we do. I don't know if that helps. There it is. Okay? Thursday. And time for 5 minutes. Everybody got one? Let's take a few minutes with this. So make sure you're not referring to any of your stored information here. This is supposed to be closed book. You don't look at stuff. Let's take about another minute.
Can we write on the backside too?
Yeah, you can. Don't make it happen. Okay. Pass them, make sure you put your name on them. Pass them centrally or centrally, What's the answer? Oh, and if there are extras, if there are extra blank ones, I'd like those back too. I mean, that'd be useful. What's the, huh? Yeah, where does it himself, what the? Okay, what is done? I mean, so, anything else that you would put in this? So there, that's what the test is for, awareness of themselves.
A mark is put on the animal, and there's 10 years, and if they approve the mark or actually 10 years, they pass.
Yeah. And it's about the awareness of themselves.
Yeah, and the marks in a spot that they can't see.
The mark says what they can't see. Others over here? Come on. Okay. So what we're going to talk about for the rest of the time is has to do with visual acuity. I mean, partly because we have very high visual, we're really good at it. But also you can see how it would actually be important in influencing, finding food and detecting predators, and seeing mates, and seeing detail in the world, and so on. How do you measure visual acuity? A lot of times you'll see that it's the cycles per degree is a measure of visual acuity, which is kind of a weird kind of units here, but here we have something that's changing fairly slowly. Here's from dark light to dark, so it's one cycle, and there's another cycle. So if that sort of one cycle takes a fairly big distance in your visual field. There are very few cycles per degree. And you can increase the frequency of the spatial change from dark to light, and so you have increased number of cycles per degree. And so it goes from low to high across the top. Okay. Now the top row has fairly dark to fairly light changes in the contrast here. So it's a very contrasty change from light to dark. The bottom row has the same level of spatial frequencies, changing from dark to light at the same frequency. So it's the same number of cycles per degree, but it's from a little bit darker to a little bit lighter. So the thing that influences your visual acuity is not just the frequency here. It's not just your ability to detect light from dark, but other things matter as well. So like the amplitude of the change. It's a very low amplitude here. So you can see that there are darker and lighter stripes here, but by the time you get to this very rapidly changing, it's maybe a little hard to see. And it's much easier to see when the difference between the dark and light stripes is more contrasted. Okay. So that's how you measure it, but it's not actually the only thing that influences visual acuity. I'm just trying to say that. So one of the things, this is probably the most important thing that influences visual acuity is the size of the eyes. This is eye diameter and visual acuity in cycles per degree. And here we are. We're at about 60, 65 cycles per degree. Along with chimps and some other old world monkeys. Okay. That's it's a very high level of visual acuity. cycles per degree, here, 65. Okay. We're not the highest. The highest are eagles. They have eyes that are approximately our size, but they have a visual acuity that's maybe more like 120, 130 cycles per degree, so double our visual acuity. But we're pretty high up there. What's considered to be legally blind is 10 cycles per degree, and pretty much everything is, according to us, legally blind, including most mammals. You know, we got elephants here who have very large eyes, but you know, their visual acuity is about 10 cycles per degree, putting them on the boundary of what is legal blindness, okay, for us. Many birds are down here below 10 cycles per degree. Some are higher. We've got some that are maybe up in the 30 range, but birds are kind of straddling this region. Mammals are you know, higher mammals and birds. Again, differ in visual acuity largely because some mammals are bigger and some birds are bigger than others. I mean, the ostrich is high up here because it's a big animal. Okay, so down here you got like mice. You have, you know, they have small eyes, so they have lower visual acuity. It's also plotting some insects and maybe some other crustaceans. So I don't know what these are, arthropods or something along here, and they're red ones. They tend to have a low visual acuity, just not able to see detail quite so much, mostly because of the fact that they're small. Okay, there's a bit of a difference between the kind of eye that organisms have and their visual acuity. We have camera eyes and they have compound eyes, and the sort of the limit to the visual acuity of organisms that have camera eyes, where you know, images focus on the back of the retina, where you have sensory cells, is due to basically how closely packed those sensory cells are, and those can be pretty close, you know, densely packed, and you know, they're cells. They're not that they're not that big, so there can be, you know, you can have very small angles that you can conceivably distinguish. In insects, they have ommatidia, which are fixed eye segments that point to a particular direction in space. And so the acuity that they have is going to be lower because each ommatidium is bigger than a cell, you know, and so it actually takes takes up a greater number of degrees as it's pointing, you know, out into space. Now, insects do a variety of things to sort of make up for that. They have, like, for example, this dragonfly has, you know, a huge number of ommatidia, and they're pointing in all sorts of different directions, so it gives them actually pretty good visual acuity for their size and so on, and for their, you know, their insects. And you can also see something else that's kind of interesting, which is that the ommatidia are not all the same size. So those that are looking up in this dragonfly are bigger. That means that they're not going to see quite as many, they don't have quite the same visual acuity when they're looking kind of up as when they're looking kind of down. You know, and this is a predatory insect, which is mostly hunting things that are kind of down below it. So it kind of makes some sense. A real extreme example of this is this fly, where, again, the ommatidia that point up, you know, where it's maybe getting information about navigation, position of the sun, which doesn't require quite as much, perhaps, visual acuity as sort of the information that it's getting about sort of like down around below it. Okay, it's like where it's getting a lot more. There's a lot more information about the location of the object. Does that make some sense? Yeah.
So big over think you can see more, or?
They can gather more light, but they're not as good at figuring out, and picking out detail. So they don't have as high a visual acuity, because each one of them is looking at a bigger angle in space. We think of ourselves as part of the reason that we have good visual acuity is that we have these foveas in our retina, which is a concentration of sensory cells in a particular spot that we focus our eyes on. Right? And so we get a great deal of ability to measure detail. In humans, the number of cells per square millimeter is something like 160,000, which is a lot. I mean, our fovea is not probably a square millimeter in size, but it's a lot of sensory cells. Okay. It turns out that among mammals, only the old world monkeys have foveas. It's kind of interesting. It's actually also those mammals that have the trichromatic color vision. Interesting. Other mammals don't have a fovea. Things like dogs have a visual streak, which is an area it's not nearly as densely packed as with sensory cells as our fovea, but it's an area of a little higher concentration. And it's a good sort of streak here. Birds have phobias. Okay, these are some birds that have not remarkable vision. You can see the fovea in there. This is round spot. The birds on the bottom actually have multiple phobias. These have two phobias. So something like an eagle, which has something like 10 million sensory cells per square millimeter in its fovea, not only has a phobia, but it's got two phobias. So that's probably why they're so eagle-eyed in terms of their visual acuity here. Yeah?
Have you spelled phobia?
F-O-B-E-A.
The circular part is the fovea?
Yep, it is. Now, who cares about visual acuity? Okay, so here are some pictures that predict the amount of information that the individuals have available to them. And we've got this, it's a picture of a butterfly, okay? And across the top, we've got different organisms. Map butterfly, reed bunting, a small bird, Eurasian blackbird, a little bigger bird, and Eurasian jay, a little bigger bird. And it shows the sort of visual acuity in cycles per degree. So pretty good here for a Eurasian jay, about half as good as ours. Really lousy for this butterfly. Okay. 2 orders of magnitude less. And this is what is predicted. Okay, so this is not a prediction of what they see. That's not what's saying here. But this is a prediction of what information is available to, like the map butterfly, if you are 2 meters away from this picture. Because of its visual acuity, it just can't distinguish particular spots. And therefore, its image of the world at 2 meters, or its image of that butterfly at 2 meters, is this. This is the data that it has to work with. Okay? Even when it's 1/10 of a meter away, it doesn't have very much of capacity to distinguish a lot of detail. Okay. This J is a lot better, although even at 2 meters, it's getting kind of blurry here. Okay. Yes?
You said this was a prediction based on?
The visual acuity of these organisms. Maybe something a little more relevant. This is a spider in a web that is being perceived by a housefly, a butterfly, dragonfly, small bird, a pigeon. pigeons that don't have great visual acuity. But the pigeon can see the spider, okay, up to 2 meters away, see kind of a certain amount of detail. The fly, even when it's, 8 inches away from the web, can't really see anything that is relevant to this. And this probably tells you a lot then about, predator-prey relationships and how, a potential prey might interact with its potential predators. This is a spider that's called Argiope. They make these little things in their web. It's called a stabilimentum, and people have always wondered what in the world they make these for. And one of the thoughts is, and seems to be borne out here, is that they make these things and put them in their web in order to make them visible to birds who then won't fly through the web. And that probably works then for pigeon. And it doesn't make it so visible that their prey can avoid them, because the flies just don't have the visual acuity to see this. So it's telling you something probably about the behavior. Okay, let's take a couple of minutes and think about what it is think about mammals and how they differ in visual acuity. So this is visual acuity on the y-axis, cycles per degree, and eye length along here. And pretty much mammals are the same. They all fall on this one line, except for these guys. These are the diurnal haplorhines, which are old world monkeys that are active during the day. Okay. This is where we belong. Okay. We're up there. We have much higher visual acuity. This is probably due to our fovea. I mean, we're the only things that have foveas also. Okay. New World monkeys, Strepsirrhine primates, are just like any other mammal. They have the same level of visual acuity based on their body size as any other mammal does. It's only the old world monkeys that have this, like us, who have a heightened visual acuity. What else do you know about things? Those organisms that are diurnal have greater visual acuity than those that are nocturnal. So that kind of makes sense. They're going to be more visual hunters. These guys they're called ephemeral, are sometimes active at night and sometimes during the day. Think lions in this case. Lions will hunt at night, they'll hunt during the day, and it's just kind of depends. And they have as high a visual acuity as diurnal. And also, those mammals that are active predators, as opposed to ambush predators. Active predators have much higher visual acuity than do herbivores. You would think maybe that herbivores would get a bonus for having high visual acuity because they're trying to avoid predators, but that apparently isn't the case. It's the predators that have high visual acuity compared to the prey animals. Okay. So there's a lot of information there that's associated with, visual acuity and the sensory systems of organisms. Okay. There's a lot more you can talk about vision. We're going to move on next time to talking about hearing. Okay. And we'll spend a lecture on sensitivity.